Okay, just a couple more orientation issues because people have been emailing me with the same questions over and over again. So as I was just talking about, the way that I have my course set up, all my courses set up is sort of module based, not assignment based. I realize that some of your instructors just have long lists of assignments, or they've actually enabled the syllabus and they use the syllabus. I don't use either of those things. So you'll notice that what you can click on, assignments, is hidden, right? For me, I can obviously click on it, but it's hidden for a reason, because I want you to access the assignments through the module, because when you do it that way, you will go directly to Connect because of how I've set up the course and paired it with the Connect section. So that's why you can't see the assignments, okay, as a list. You just can click on them, right? And then it's going to take you, should take you right to the assignment. And it looks to me like most of you have at least gotten into chapter 1 assignment and worked on it a little bit. So I feel like most of you probably are doing okay with that. Someone asked about the virtual lab, right? Those are in Connect. And the challenge with them is you have to keep doing it until you get 100, right? It's like the other Connect assignments, in that, as it stands right now, we don't have the ability to change that so that you can have a score for that particular lab exercise. The virtual labs are going to be, they've expanded them, and most of them are like literally brand spanking new, like as of November, and I haven't looked through all of them yet, but we're going to start incorporating them until we get the lab kit. And then after we get the lab kit, I am going to add more virtual labs than there are right now, because in the fall, there were only a handful of virtual labs available, actually the ones that I have in here. But now they've added several more. So I tend to put the lab exercises toward the bottom of the module. I put this one early because it's kind of, to me, it feels like it's part of like the introduction to the course. But normally I will put the PowerPoint first, or a note outline if there's one available, the video lecture of the chapter, then the homework with the little pen here, right? Those are homework assignments. This is just a file. This looks like this is a page because it's an actual video. And then again, then we start over 4, chapter 5, and then usually the labs are at the bottom. The virtual labs are at the bottom, and then the lecture quiz, if there's going to be one in that module, it'll be at the bottom. So there's no lecture quiz for the first module because we're just all kind of still getting logged in, and I'm still actually getting students added to my section. But the first lecture exam has been set on February 16th. It will be open all day. The exams are always on Tuesdays, and they're open from 12:01 a.m. until 11:59 p.m. Okay, you can't see anything when you go in there now, I believe, except the box that tells you how many seconds you're going to have for each question, and just a little bit of the introduction, but you can't actually see the questions yet. You won't be able, those won't go live until whatever that says, February 16th. I know that lecture quiz is long. There's 73 questions. It will include chapters 1 through 5 plus 8. Now, chapter 1, we're going to go over today because I don't have a video of chapter 1. Looks like I only have PowerPoints for whatever reason. Chapters 2, 5, and 8 should be all review for you from Bio 1. If they're not, you're going to have to spend some extra time studying those chapters. But don't spend all your time studying those chapters, because the amount of questions from each chapter is about the same, except chapter 1, of course, because it's kind of like, hey, welcome to microbiology. So the new material is really a little bit of chapter 1, chapter 3, not even the whole thing, and chapter 4. Those should be the new material for you to study. The rest is review, which is good and bad. It's hard to study stuff that you think you already know. I understand that. That's why I've made the Connect work for chapters 2 and 5 and 8 rather long, because I know that you're not going to want to study it. You're going to look at it and you're going to go, oh yeah, I remember everything about the Krebs cycle. Oh, I don't need to watch that video lecture. And then when you actually get the questions, you're going to be like, oh crap, I don't really know this. So the connects are long for those chapters where you have to, that it's a review chapter. And I'll try to break some of them up a little bit more, but I get it. They're not always pleasant to do, but, and they are a lot of work, but this class is a lot of work. And if you do the work, it will help you get a better grade. The dates will be changing on the assignments. So I wanted to explain that once rather than having to type it 40 times or 60 times. So what happens is when I set my course in Connect and I copied it from the fall because I wanted to use all the same assignments and I want to add some assignments, it asks me, do you want to reset this course for a new date? And so I could put in, start the course January 20th. And so it took all of my assignments from fall and it decided, based on how spaced out they were in the fall, what dates they were going to be due. And clearly they're not correct, okay? So I'm going to be going through and changing, not the assignments that you've already started, but the assignments that haven't yet begun. Those dates are all gonna change. Please do not try to go into Connect and do this assignment before it's actually available, right? I think I've kept them all hidden, except I think chapter 3 I may have made live, but I am going to switch this date. It's not going to be due February 8th. It's going to be due before that. I'm undecided. In the past, when I had the course as a live course, as an in-person course, I used to make all the Connects due the day before the exam, so that students could use the Connect or not use the Connect when they were studying as they pleased. And I'm leaning toward doing that again, rather than have individual dates scattered throughout this whole module for when the actual homework is due. Now, the disadvantage of that is if you don't do it until close to the end, it's not gonna, you're gonna be screwed. I mean, you're not gonna be able to get it all done in one day, and you're not gonna be able to get it, and then you won't have any more study time left to study for the actual test. So I am kind of undecided about that. I'm probably going to make the Connects, I think I'm going to break them up and make them do the Monday before, like on a Monday of each week, just to kind of help you guys pace yourselves. So that's probably what I'm going to be leaning towards, so you can keep an eye out for those changes. But I left them all till the end in the spring and the fall, and it didn't work very well because a lot of students waited until Monday, like Monday at noon, to try to do 8 Connect assignments, and some of them are over an hour each, and they were freaking out. And then they didn't have time to study, and then they complained that, and I'm like, it's not my fault that you waited till the end to do them, but I'm going to try to make that so that you can't wait to the end, to help you pace yourself in the course and study as you go along. The virtual labs, I probably will make those all due the Monday before the test. I would prefer that you do them sort of as you go along during the time, but I get it. You've got a lot of other stuff you're doing too. So again, expect all these dates to change, especially in module 2. Module 1 is pretty much set. I kind of have to leave it the way it is because I had to open those assignments to be able to put them in the module. The rest of these are here, but they're, like I said, they're going to change dramatically. Obviously, the lab isn't going to be due after the test, right? This will be the test, the exams, lecture quizzes, they're called in Connect, in Canvas. They're always at the bottom of the module. And how I have it set up is each module ends with a quiz, right? So you can see this one's going to be 3 chapters plus a little bit of chapter 3. This one, oh, those are not supposed, yeah, this one has a short answer section that's a separate quiz. This one is a lecture quiz, 3 chapters. This one, I think this one was just 2. This one, this was the, let's see, there it is. I don't know why it's up high. I think that might have been the final. These 2 were the final, the 2 quizzes, and I just did them on 1 chapter each. So I'll probably change for the spring. And then the lab modules are down here. This is for your lab kits. This is all of the introductory information with the Carolina kits. This module contains the actual Carolina documents that you're going to need to read to be able to do the experiments. They're going to be videos that I've done. And then there are also, when you open up these Carolina documents, they contain, let's see if I can show you one of them. They contain, see how they have these live links? And then you can actually go to, actually this is the supplemental one, sorry. Go back. There's some supplemental videos in there in the lab stuff, but there are also, Carolina themselves also puts out videos, and you can watch those here, the lab kit experiment, this little document. When you go there, it previews the document for you. Let's see if we can actually get there. And it'll show you, like, this is the booklet from Carolina, and this is step by step, how long it's going to take you. This is safety equipment you need, this is the background, and then the procedure. When they get to the procedure part, it's all background, background, background. When they get to the procedure part, usually what you'll see is a little proper hand, one of these, and these are the Carolina people telling you how to disinfect, how to dispose, how to make a surface disinfectant, and then how to reactivate your lyophilized culture. Anything that you actually need to do, there'll be little videos of the Carolina person doing that. And then, as I also told you guys in the video last week on Thursday, I have also created lab videos, and mine are by the day, because when we did it last semester, we need to complete all of the lab kit in about 2 weeks. The tricky part is we'll need to do that again, because once you start growing bacteria, they don't live forever. They only live on the media really for about 4 or 5 days, maybe a week. So you need to grow them, and then once you've grown them, you need to do the experiment that you're supposed to do with them, and then we need to use them for another experiment. So don't throw your cultures away until you are all finished with the labs. And so you'll see these videos that are labeled Lab Kit Video Day 2, Lab Kit Video Day 2, Lab Kit Video Day 3. That's me going through the actual labs that you're supposed to be doing in the order that I thought was the best way to get this done, okay? And I think they only go up through 6, right? The lab reports that you're gonna turn in, you are gonna turn in one lab report about the microscopes and using the microscope, 1 or 2 labs for the microscopes, and then I'm gonna split this lab kit. It was one giant lab report. I'm gonna split that into lab reports number 2, 3, 4, 5, and 6 based on the exercises, okay? So it'll be a little bit less of the writing up that you'll have to do for each of those labs. And then those will show up as assignments like this with a little pen on there for you to actually turn them in on Canvas, in Canvas. All right, so I wanted to talk about that. And then I wanted to just, I wanted to remind you, I still don't have any more information about the lab kits. When I get the information, it'll pop up in an announcement. So I would encourage you to check your announcements every day for this class, right? Again, on the home page, the 3 most recent announcements are going to be on the top. And then if you need to see more of the announcements older than that, you need to go to the announcement section, okay? When I get lab kit information, which I am guessing I will be getting it probably next week, right? First week of February, I'll probably be getting the information. Again, remember, I'm going to be turning in your participation grades, your /attendance grades, not at the Zoom meetings, but your attendance by going into Canvas and doing your work. I'm gonna be turning those in, and after I turn those in, then that will generate the finalized list of who gets a lab kit. I'm not sure how they're going to give you the lab kit information. They may just send you an email with your code in it. Last semester, they did ask students to come to the bookstore and pick up the physical code. Once you get the code, it's actually a voucher, code for a voucher. You will go to the Carolina website, you will redeem your voucher and give them your address, and they will send the kit to your house. Please do not give them an address that you can't access. that you are not going to be able to go to in case someone gets COVID, right? Don't give them an apartment address if you're going to be moving in the next 2 weeks, right? So try to think ahead a little bit and give them a valid address, because you're only going to get one kit, right? And then it's going to be on you to be sure that you get it and open it up. When you receive it, I'm going to give you, there's a list of all of the items that should be in the kit, and you're going to need to check for things that are broken or missing, and then you'll need to contact Carolina and they'll send you the replacement items, okay? So, and also make a little space in your refrigerator in your house, because some of the items do need to be refrigerated. And again, we're going to refrigerate them and use them maybe 3 or 4 weeks from now. So they need to kind of stay in your refrigerator for a while. The things that are being stored in your refrigerator do not contain live bacteria. They are mostly just the media. You will need to have a place where you can reconstitute your bacteria and grow them and keep them out of the reach of children and pets. And obviously other people in your household. And last but not least, if you want to make an incubator, I think I mentioned this in the first video. I had a student last semester who made a homemade incubator with a light bulb and a wooden box. I will put those plans up probably in the first lab module. And then you can, she submitted them to me last semester, and then I saved them and I'll put them up there so you guys can see or modify that if you feel like you want to make an incubator. Okay, does anybody have any questions about generally about the course? Because now I'm going to start talking about chapter 1 and I'm just going to go through the whole chapter. So if you have a particular question about the course, how the course mechanics, I'll certainly take that question now. But if you don't, then I'm going to move on to chapter 1. Yes, no, maybe so.
I actually have a question. I'm sorry.
Okay, that's fine. No, that's the point. You get as many questions as you want, Janice.
So I just, because you have in some modules posted like notes or videos and stuff, for some chapters, but not for others. So I was wondering if some weeks there's going to be like 2 chapters due, but only 1 lecture because we only meet on Tuesdays.
Oh, so my plan was to meet on Tuesdays with you guys for answering questions primarily. Today I'm giving you a full lecture because I don't have a video recorded of chapter 1 for apparently from summer or from fall. So there isn't a lecture for today like that's already loaded, right? But for example, for chapter 2, there's not going to be a video because that's just a review on your own chapter. Chapter 3, there's no note outline. So I'm expecting you to watch the video and take notes, right? Occasionally there's note outlines. So there's one for chapter 4. Looks like I've got one for chapter 6, and but not for 9. So if there's no notes that are posted, then I'm just sort of expecting you to watch the video of the chapter and make your own notes. Right, and you're right. So for this week, for example, or for this module, right, it's going to be just mostly chapter 1 and then those the homework for chapter 2. But for starting with module 2, which if you're kind of planning it out, which I didn't do it for you because the microbiology students usually like to be a little more independent, right? So ideally, I would plan to, on Tuesday, say, of next week, or before Tuesday of next week, I would watch the video for chapter 3, maybe do the homeworks, look at the PowerPoints, watch the video for chapter 4, and then next Tuesday when we meet, I'm going to ask you guys, does anybody have, focusing particularly on chapters 3 and 4, does anybody have any content questions or things that they didn't understand from the homework, or things that are missing, or, you know, what did you mean when you said this or that in the videos? I'm not planning on doing live video recorded lectures again, because this would be the 3rd time. So I'm just going to post the ones that I already have. I mean, obviously they're already in here. So some chapters are really long, like chapter 4, there's 2 videos, right? But there is a note outline to kind of help you. And then there'll be homework, but again, this date's going to change because that homework is not correct. Chapter 5 is a short one, so there's just 1 video for that one. And then chapter 8, super long video, some PowerPoints, and then homeworks. And this was a Q&A from the fall. I'll probably leave it sitting there if you want to watch it. And then when we meet on February 9th, I'll be asking, does anybody have questions, material, worries, concerns from chapter 8, right? So like every Tuesday, when we meet in our Zoom meeting live, it'll be your opportunity to ask me any questions you want about the content for the upcoming exam, basically.
So the meetings aren't necessarily for lectures.
Right.
Okay.
Right. I think chapter 1 and chapter like 16 are the only ones that don't actually have lectures for. So I don't know what that is. So some of them, but for the rest of them, they're kind of all in there. And other than this first module, which because you have to have kind of like a, hey, this is how we're going to do this class module. Other than module 1, each module has everything that you need to do in it before the test.
Okay.
Right, so you can kind of pace it out however you want. Some students are already asking me, questions from chapter 8. And I'm like, whoa, just let's slow down a little bit here. We're not there yet, right? Let's kind of, so every Tuesday when we meet, I'm not necessarily going to give you a full lecture again, but for example, since chapter 2's in this first welcome module. So for example, if today someone had already looked at and read chapter 1 and looked at chapter 2 and they were just like, how much of this chemistry are you going to ask us on the test? And I would answer you, and then you would say, okay, can you show me how to make an ionic bond again? Then during our Zoom meeting, then I am going to actually give you a lecture on that topic that you ask about specifically.
Mm-hmm.
That makes sense?
Yeah.
But I'm not going to go and give lectures again for all the things that are already recorded, because it doesn't, they're up there. You just have to watch them. They're boring, but you just have to watch them.
Okay. Well, thank you.
You're welcome. All right, does anybody have any other questions, like general questions like that about how the course is sort of going to work? You'll be excited once we get the labs. I don't have my little microscope in here, but they come with a little baby microscope that's like this tall. So you can look at things under the microscope, which is kind of fun.
Actually, I'm sorry. Okay, one more question. So like this week you have stuff due Tuesday and stuff due Thursday. Are most weeks gonna be like that? Or is
yeah. I'm like I said at the beginning, I'm undecided. I used to put them all the connects due right before the test. And then students didn't do them until right before the test. And they got into trouble doing it that way. So I have not gone through and reset all of the connect due dates, all the homework due dates, to coincide perfectly with the quizzes. So I'll be working on that this week, quite honestly. Most likely, you'll also see these change. I'm probably going to put a header here before chapter 3 where I'm going to call it homework. So I'm going to probably do that right now because students are having trouble. So, right, so I'll do that, homework. And then you'll know that that's a connect assignment.
Okay.
Right. So that I can't spell correctly. There. Homework chapter 3 update. Okay. Right. And then you'll know that, oh, that's something I have to go to connect and do. The labs I'm going to be labeling virtual lab and lab kit. Right. And then anything that's labeled video is a lecture, basically. And yeah, and they're all open. Like you can just click on them. They're all in YouTube. I did not edit them, so they're, you know, they just kind of are what they are. And this one won't let me see it because there's too many people on Canvas right now. Let's try a different one. Let's try a short one. That one should be kind of short. Oh, let me see it. Yeah, it's too many people trying to use the same stream right now. But hopefully you guys can see it. All right, then, does anybody have any other questions? Because if not, I want to start talking about chapter 1 real quick here. Because there's not a lot in chapter 1, but there are some things that you do need to focus on. Yes, no, okay. Then I'm going to share these PowerPoints with you while I'm talking because I don't, other than that, I don't have any pictures, right? So chapter 1 in all books is always kind of, you know, welcome to the wonderful world of microbiology. And these are all the things that you could possibly learn about if we had a whole year for you to learn about microbiology. And I'm actually going to start with this one because this picture, because this goes through what are the different types of organisms that we're going to be studying in micro. And this is like one of the opening pictures in your book. I wanted to remind you that there are basically 2 types of organisms that we're going to be studying. One of them we refer to as acellular because they are not cells, right? So those are prions as well as viruses. We study viruses in microbiology because they infect every other type of living thing. There are viruses for bacteria, for fungus, for protists. They infect all kinds of cells, plants and animal cells, but they themselves are not living things, right? And we'll talk more about why they're not living things when we get to chapter 6 and talk just about viruses. The other side of this diagram here are cells that we're going to be discussing. And these would include, of course, bacteria, because when everyone thinks of microbiology, the first thing they think of is, oh, we're going to study bacteria. We're also going to be studying eukaryotes because fungus, and mostly protists, other protists, some that are pathogenic and others that are beneficial, are also considered microorganisms. Take a look at these little gray circles here. This is supposed to give you some sort of a size difference, right? Because we're going to be thinking about using a microscope. It's microbiology. So biology, study of life. Microbiology, study of small life. So these guys, the prions and the viruses, we can only see those with electron microscopes. Really, really powerful microscopes. And the prions, we have to use even a special type of an electron microscope to see them. And then notice that about these bacteria, look how much larger the bacteria are. And you'll notice they have a size marker there at 1,000 nanometers, which is equivalent to 1 micrometer. And a micrometer, remember, is 1 times 10 to the minus 6th meter. So it's 1,000th of a millimeter. And then look how large the eukaryotic cells are. We say they're usually about 10 micrometers in diameter, right? So the eukaryotic cell is like ginormous compared to the prions, the viruses, and even the bacteria. It's about 10 times bigger. Now, unfortunately, the microscopes that you're going to get in your kit are only going to magnify things up to 400 times the size of life. So if you used a real microscope in Bio 1, for example, it would be equivalent to using the 40x objective with the 10x ocular magnification. So we are going to have a hard time actually seeing the shape of the bacteria because they're going to be so small, but we will be able to see some other interesting things. So these are the types of organisms we're going to study. And I would expand this even further. So this picture is also in your textbook, and it shows all of the different organisms we're going to be studying. So this is a prion. This is just protein tangles in someone's brain or an animal's brain. A virus, of course, some bacteria. And then these, of course, are all eukaryotes. So we've got some protozoans, fungus, And of course, a parasite, this would be an animal, right? This is platyhelminthes. We're going to be either studying each of these organisms because they cause disease or because they are beneficial and they provide mostly decomposition as well as providing, they put a non-photosynthetic protozoa in here. If we had algae there, we would say, because they're going to be providing oxygen for the rest of the planet. Now, these different types of microbes, these different types of microorganisms are studied in different sort of subcategories of fields of microbiology. And so I did want to run through these a little bit to give you a little bit of perspective, because again, I think mostly when we think about, oh, I'm taking microbiology, we all think we're taking a medical microbiology course. Not necessarily. Okay, this is a general survey microbiology course, and we're going to touch on all of these different sub areas of microbiology, or sort of like sub-disciplines. I can make this bigger. So we have, there we go, I can make it a little larger. So first off is medical microbiology, and we see a person in here, a lab tech in an actual research laboratory, right, looking at bacteria, microbes, unless we're saying microbes, so it would be bacteria, viruses, protists, animals that cause disease in humans and animals. And this is really only about very small percentage of the microbes that are out there. It's usually, we say it's about 150 bacteria species that actually cause diseases in animals and humans. And there, of course, are thousands and thousands of species of bacteria that we know of. Public health, microbiology, and epidemiology. This, I think, doesn't need much explanation in the current environment of COVID-19. This would be involving, of course, education to hand wash, wear masks. This is how you can prevent disease. Then this is how you can control the disease. And of course, this is how you can monitor the spread of the disease, right? Contact tracing is part of epidemiology, as well as reporting case numbers and treatment options and vaccine numbers, right? And vaccination numbers. So you see some people out in the field there talking to some people about different types of ways to prevent spread of infection. Looks like cholera, it says, right, in Haiti, right? So that's always beneficial. Immunology is a subcategory of microbiology. So this studies how the human, particularly the human immune system, responds physiologically to a microbe. Okay, so we have about 3 or 4 chapters about immunology later in the semester. It's my area of expertise from a long time ago. It's what I have my PhD in. Industrial and obviously that's going to overlap with medical microbiology. Industrial microbiology, I think, is something that a lot of students don't think about. So these are procedures and processes that protect our food and water. So think about the whole food chain. This is some chickens that are being inspected, most likely for pathogenic disease before they are finished being processed and packaged. This also includes things like, if you go to GNC, right, and all of those supplements that are all over the walls, many of those are actually produced in a bacteria or a fungus and then purified from a bacteria or fungus. Amino acids, of course, some things that we eat, like anything that's fermented food would be considered industrial microbiology if you can buy it at the grocery store and you're not making it at home. So things like yogurt, cheese, beer, most pharmaceuticals, vitamins, all isolated from bacteria or yeast. And we call it industrial microbiology because it's going to have to be on a huge scale in order to grow enough bacteria or grow enough fungus to actually be able to purify these compounds they make in really tiny amounts. Overlaps a little bit with agricultural microbiology, but this does focus on domesticated animals and crops and plants that we use for food, crops, right? So you'll notice there's plants specializing on diseases, soil fertility, and nutritional interactions, and the animal specialists, of course, working on infectious diseases associated with animals that are basically herded animals or chickens, things that we use in our domesticated that we use for food. Microbiology, like of our pets, kind of overlaps a little bit with medical microbiology and a little bit of agricultural microbiology. Right? And then last but not least, we have environmental microbiology, and this would include all of the organisms that people study that are in the naturally occurring in the water, soil, rocks, in the Earth's crust, and even in the air or in outer space. Right? So you see this guy sifting here, collecting some microorganisms, probably from the silt in the lake, so he can determine what maybe there's a disease that's affecting fish. So they would have to know what the organism was and where it was living in the natural environment, and then how the fish are getting exposed to it. So again, a lot of these types of microbiology, they cross over, and people from several areas might be working on the same project, right? So for example, like right now, I would say all of the efforts on COVID-19, we're having public health microbiologists involved, we're having medical microbiologists involved, we're having immunologists involved, and I would even go as far as to say we're having industrial microbiology involved as far as the vaccine production, right? So a lot of times you have many of these interacting to work on a single problem. And we talked about the kinds of organisms that we'll be studying. We talked about this. Okay, so let's take a look a little bit about sort of the history of microbiology, right? Because I'm an old person and I think history is interesting, right? So this is a picture of a microscope that Anton von Leeuwenhoek is a replica of a microscope that Leeuwenhoek made in the 1660s. He lived in Holland and he was a cloth merchant by trade, but he wanted to see, basically, first he wanted to see the fibers of the cloth so he could assess the quality of the cloth. So he basically had a little magnifying glass that he carried around in his pocket. And he was so interested in looking at things that you couldn't see with the naked eye that he kept working on making this lens smaller and smaller, and then attaching this little, almost like a metal toothpick, so that he could put a specimen on there and then look at it with his lens, right? And we all know the first thing he looked at was he scraped his teeth. 1600s, no one's brushing their teeth, right? And he saw some different types of organisms, and I was just going to draw them for you real quickly. He actually saw, right? He actually saw this, so sort of a rod-shaped organism. He saw some round cockeye-shaped organisms, spheres. And then the one that is most interesting to us, of course, is he saw one that was kind of zigzagged in shape. That's a spirochete, and you have them in your mouth, even if you don't have syphilis. That's the most famous spirochete, Treponema pallidum, but you have them in your mouth. And then he also, when he was studying, he also saw in his mouth a bacteria that he put a little dashed line because it moved, right? So he had seen bacteria with flagella before we knew what they were. So that was probably one of the most important sort of advances in microbiology, or the beginning of study of small life, is to be able to actually see the small life. And then, of course, let's make this a little smaller so I can go to the next panel. And then we have this experiment here in the 1800s with Pasteur, looking for how we could prove that life was not spontaneously just poof coming into existence, but it was dependent on previous cells. So Pasteur's experiments to disprove spontaneous generation, these are taking place almost 200 years after Leeuwenhoek. And the general idea, I'm sure most of you already learned this in Bio 1, but I'll just quickly review. Spontaneous generation was the idea that you could put together certain animate and inanimate objects and create life. So for example, there were recipes. So like if you put cloth and corn and a bushel basket upside down in a dark place, those are three inanimate things, inanimate objects, you would be able to spontaneously create mice, right? In 28 days, because that's how long mouse gestation period is. And there were other recipes to get different organisms to appear spontaneously. And there was a whole group of scientists who, through just inductive reasoning, didn't think that was true. And then using deductive reasoning, were trying to show that it wasn't true. And those experiments began in the early 1700s with Reedy, if you know of those experiments. And there were not hundreds, there were probably at least 100 experiments between Reedy and Pasteur. But Pasteur gets the credit for just proving spontaneous generation because of how he constructed his experiment. So his experiment was, by now people had seen microbes, right? And he had bacteria in a flask, and he boiled the broth to kill them, and then he bent the neck of the flask. So these flasks are called swan neck flasks. It's really hard to say fast, and because they have a neck that looks like a swan. And the idea is that the flask is open to the environment. So if an organism would happen to, from the air, would happen to land in this tube, they would be able to land here, and maybe if they were motile, or if they had a flagella, they might be able to migrate up this tube a little bit, but if they can't get to the food, they can't make more microbes, right? So right now this broth is sterile, meaning it's free of living things, and he was able to hold this broth sterile for about 3 weeks. And we know it was sterile because it's crystal clear. Broth that's infected with bacteria becomes turbid or cloudy almost immediately, within a day. And in order to demonstrate that the broth was still capable of sustaining life, that's when he broke off the neck of the flask and showed that if bacteria or fungus could get into the flask, they would be able to contaminate it and be able to grow, right? But as long as the neck was intact, the broth stayed sterile. So Pasteur said, you need cells from somewhere, from the air, from the environment, in order to make more cells. And then we know that Schleiden and Schwann in Germany, which is where the better microscopes are being produced in the 1850s as well, they're finally compound microscopes, so you can see higher magnification, and more importantly, you can see things more clearly, more distinctly, with better resolution. Schleiden and Schwann, one with plants and one with animals, started to describe the word cell, right? And they came up with the cell theory. They proposed the cell theory that all living things are composed of cells, and cells are generated from other cells. So the cells divide to make more cells. It's not spontaneously formed from nothing, which dovetails very nicely into Pasteur's experiments, obviously. And we just list a couple other big milestones in microbiology. Here's a picture of Lister spraying phenol over a surgical area to disinfect, right? So that was a big advance, was being able to disinfect both surgical equipment as well as finding antimicrobials, substances that would actually inhibit or kill the bacteria in people. A man named Robert Koch developed what we call Koch's postulates, and these are a series of steps to be able to determine which organism causes which disease, which was very prevalent in the 1800s. And then I did want to list here, these are some of the major advances in biotechnology, right? Starting in the 1970s, being able to manipulate microbes on purpose with very specific activity, like being able to cut a chain of DNA in a microbe and replace it with something else. We would call those restriction enzymes, and we would say this is the beginning of biotechnology. PCR, in order to be able to detect various sequences of DNA. And now in the case of COVID, we use a PCR test to detect the SARS-2 RNA. And then the importance of small nuclear RNAs, right? This would be the CRISPR, as well as other nuclear little tiny RNAs that actually turn genes on and off that we were unaware of before. So we could talk for days about the history of microbiology, but I'm just going to leave it at that. This is just a slide to remind you the differences between prokaryotic cells and eukaryotic cells. Again, that should be something you know from Bio 1. All the prokaryotes are mostly single cells. They lack membrane-bound nucleus. They lack other membrane-bound organelles, and they reproduce by binary fission. I would add their DNA is usually circular. It is a single chromosome, so they have a lot less DNA, but they do have DNA. should actually put that on the slide, but I have to edit it someplace else. The key for chapter 1 here is notice that I've included both domains of bacteria and archaea. So we have 2 domains, biggest groupings of living things that are prokaryotes, and 1 domain, eukarya, that's all the rest, right? And again, these are characteristics that you would know from somewhere in your past. And then I did put the picture of the person with doing her inductive and deductive reasoning, right? Where she's observing something, and then that's coming to a general principle, and then she's actually testing the principle and finding out that yes, when you open up attachments on your computer from unknown senders, it can give you a virus that causes your computer to crash. Don't do that. And then this panel I put on here to remind us of how the organisms are all classified, which I probably should have put before I put the woman with her inductive and deductive reasoning. This is sort of the current web of life that we have. Notice we call it a web of life more than a tree of life now. Notice that the bacteria and the archaea, there are a lot more of those and a lot more diversity of bacteria and archaea than there are of eukaryotes. 3 kingdoms currently, 5 kingdoms established in bacteria. We'll talk about them a little bit more in chapter 4 and again later. 3 kingdoms in archaea. And then 5 kingdoms in eukarya. There are a lot of other, notice, right, like 3 kingdoms, and we've got all these different differences here. I'm trying to keep it simple, right? We call the people who do this taxonomy, we call them the lumpers and the splitters. The lumpers would say 5 kingdoms, 3 kingdoms, 4. The splitters would have as many as 13 kingdoms in eukarya. Right? So we're still in like 200 level class, so we're trying not to overwhelm you. I would just remind you of the classification, the order of classification. This is the largest group, of course, and then we get smaller and smaller and smaller. Most of the organisms that you'll see in the text are going to be defined by genus and species. They will be italicized, right? If we were not able to write in italics, if we were in the classroom and you had to actually physically write things down and turn them in, be aware that you would always have to underline the genus and the species name with 2 separate lines. That's proper scientific nomenclature. I listed here that almost always bacteria are going to have a strain after them or a subspecies, and there's usually a comma here, and then it's usually a series of numbers and letters, and it often has to do with where this particular subspecies was isolated, where and what lab and what isolate it was. Like sometimes it'll have a number like 178, and that means it's the 178th patient that they isolated it from. The E. coli that causes hemorrhagic diarrhea has a subspecies name, just like we would MRSA for example, would be a strain or a subspecies of Staphylococcus aureus, right? And I think that's pretty much the end of the chapter. All right, I am going to end my recording because I'm almost out